And before I turn it over to our speakers, we have a great array of speakers. We have protocol co-chairs, Craig Hendrix and Mark Marzinke, who will be presenting, and also following them will be Marianne Gilday, who will be talking on the clinical operations side of HPTN 106. So we have really great presenters lined up for you. But I wanted to just set the stage a little bit. You're going to hear a lot about the douche and what kind of led up to this douche and what's going to happen next. But I wanted to make sure everyone remembered all the advocacy that has gone into the rectal microbicide field. IRMA was a national or international coalition of advocates pushing forward for safe and effective and acceptable rectal microbicides. I co-created that with my colleagues, Marc-Andre LeBlanc, J.D. Davids, and Kim Mulgee 19 years ago. So in 2005, we launched that. And this image here you see on the right of your screen is Tia Irma, who we had a South American chapter in Lima, Peru. If anyone's here from Lima, hello, Lima. And she was our Irma mascot in Lima. And Lima, as many of you know, took part in some rectal microbicide research. And I just want to share some of pictures from my scrapbook. I'm not going to describe each of these, but Irma did a lot of different things. We helped collect rectal douche and enema data. We did lots of outreach, created campaigns to draw attention to rectal microbicides. We worked on a project called The Rectal Revolution is Here, which was a video for clinical trial sites that we created with Population Council. In the left-hand corner there, you'll see the facilitator's guide in Thai. That video was in Thai, Spanish, and in English. So did lots of things to try to draw attention to the need for rectal microbicides. We also worked on lube access. We put out reports. Love a good sticker, so our rectal pride stickers were very popular back in the day. We created we did a whole project or created a project that focused on Africa and Africa's need for rectal microbicides, because it wasn't that long ago where people were actually saying that we don't really need a rectal microbicide in Africa because, first of all, there aren't gay people or people who have anal sex, and straight people don't have anal sex. Those were actually things that people believed 15, 20 years ago. And so these are things that we push back on as advocates, working with the International Rectal Microbicide Advocates. And finally, I just want to call this out. The Microbicide Trials Network, which sunset a couple of years ago, was the home for all rectal microbicide research. And really carried the torch forward, really helped create not only created the field and moved it forward and got it to this place now where the HPTN, thank you very much, is taking on rectal microbicide research and moving forward this phase 2 study. But this image here is of a wonderful document called the Pleasure Principle, which was developed by MTN and our good friend Claire Collins, who used to do communications for the rectal microbicide portion of the MTN. This kind of walks through some history of rectal microbicide research and advocacy, and I will drop the link in the chat. It's a couple of years old now, but it's still really great to sort of see where all this came from and hear about some of our originating scientists who really forged the field, people like Ian McGowan and Peter Anton, Alex Carballo-Dieguez, and Craig Hendrix has been in the mix, in the rectal mix, since the very beginning. So thank you so much for that, Craig. Now, without further ado, I am going to stop sharing my screen, and I am going to go ahead and invite our first speaker today, Protocol Co-Chair Mark Marzinke, who is with Johns Hopkins and obviously the HPTN working on 106. So Mark, you can go ahead and share your screen and get started. Thank you so much.
Great. Thank you so much, Jim. I think what we're going to do is switch the order because Craig has a nice historical perspective on some of the things that you've presented, and I'll follow him. So Craig, if you want to pull up your slides.
Thank you for that. I should have remembered that. That's what our plan was, and I got all excited talking about the pleasure principle, and I lost track. So, Craig, you are first. Go ahead and share your slides.
All right. Can you hear me and see the slides okay?
All are great. Thank you so much.
Perfect. And everybody should know this is Jim's favorite topic. So he's gonna get excited. He's probably gonna tear up at some point. You might see him jumping up and down, but you know, that's the Jim we know and love. And boy, it's lovely to see Tia Irma there, a blast from the past. And I know she's with us. I know she's with us. So are you all ready to rev up? I'm just going to give a short introduction to sort of how we got to HPTN 106 and then turn it over to Mark, who's going to talk about more of the specific background and talk about the protocol itself. And then we'll move on to Mary Ann. And please put your questions in the chat as Jim has already invited you to do. So these are my disclosures. Very big picture. We've got a lot of drugs that are available to us for development, for both treatment and for prevention of HIV. And the way this begins has been with treatment, where treatment has picked up quite a number of these different strategies of different antiretroviral drugs, which have then been moved into prevention. And we currently have many drugs licensed, but the backbones for the most part are the two nukes and integrated strand transfer inhibitors, among many others. And I think we're moving very rapidly to an age of 2 drugs back from 3, which is a terrific advance. The prevention strategies that we all know now are driven largely by the same drugs and the same formulations, and sometimes different regimens. And this is an important difference. But these are largely repurposing the non-nukes and integrase inhibitors, and now, of course, a capsid inhibitor. And I only have that in parentheses because the FDA hasn't put its stamp on it yet. I'm sure it'll be a brief matter of time before that's under consideration. And these are all highly, highly effective, better and better and better each time this is looked at to provide systemic and a retroviral pre-exposure prophylaxis for HIV. And repurposing from treatment to prevention is straightforward. This is not to say that it's easy, because there's nothing easy about it. But what you do see is that there's many lessons learned as trials designs are used and these evolve. And what you see, if you look just above the box, is that the time from sort of the beginning prevention studies to actually having proven prevention formulations and products is shorter and shorter and shorter. The questions primarily in the development of which drugs to drop. We go from 3 drugs to 2 or to 1. Which doses to reduce, and once PrEP was rolled out, it in a short number of years went to the 2-1-1 on demand or event-driven strategy. And once the drugs and the doses are chosen, then you complete the phase 3 trial, which is a ton of work. Microbicides, in contrast, are hugely more complex, largely because the dosing strategies in a rectum or vagina are very, very different than pills or injections. So I'm going to look back in history and show you that for the vaginal microbicides, that development actually began within a year or so of the systemic antiretrovirals being used, studied for treatment, and soon thereafter licensed. Originally, they were long-acting gels, and then those gels, none of which proved to be effective after many large randomized controlled trials that were global in scope, transitioned into antiretroviral gels. And then when those gels, I'll go a little bit more into that in just a moment. And now we have a whole variety of fast dissolving films, vaginal inserts, and vaginal rings. And the very exciting part of all of those is that many of these are being considered as multi-purpose prevention technologies that will be useful for, among other things, HIV plus STIs or HIV plus pregnancy prevention, contraception. So very exciting developments on the vaginal microbicide front, especially for MPTs. The rectal story comes along a lot later. The very first federal investment in grants was from the CDC in 2001. And we see these, as you see this repeating theme, the antiretroviral gels came directly, in fact, the very same products that were being studied, had been studied in women for vaginal use, were being studied in men that had sex with men and eventually in transgender women to see how those would work. And we've looked at quite a number of these, and I'm going to summarize these quickly in the next couple of slides. The point I want to make here is none of these products over this 23 years of development have gone far enough and have not really been ready enough to go to a phase 3 randomized controlled trial. And I think that there's really no way to do simpler PK bridging kinds of studies because the application to the rectum is so much different than taking a pill or getting an injection and having the drug go essentially everywhere that it goes, as complex as that is. So there have been a number of questions that were important to answer, and we are still trying to answer some of these. Is the candidate drug product protective? Is it antiviral for HIV? What is the anatomic target for drug action? What is the drug concentration and the duration of the drug concentration at target that would be protective? Are there any behaviors or products that might alter the drug distribution or its effect, especially an issue when we're talking about topical application, rectal or vaginal? Are there any other drugs or vehicles or sex products that might actually be toxic in some way and make it more difficult for these microbicides to work? And then really most important is if we develop it, will folks use it? And I think I'll try to make the point that we've learned a lot from the vaginal program, which continues and does have a licensed product in some parts of the world, the dapivirine vaginal ring. But we'll contrast that because these were developed at different times before and after many of these questions were actually answered. The tenofovir gel was developed and actually was studied in 3 very large randomized controlled trials. These were all international trials. These began after well-designed phase 1 and phase 2 studies. The Caprisa 004, which you see here on your far right inside this red box, and it's red because no NDA was ever submitted. There was no application to the FDA or any other regulatory body for licensure of this vaginal formulation tenofovir product. And in the Caprisa 004 study, it worked kind of okay, 24 to 39%, but it worked pretty well, 73% in post hoc analyses if you adjust for the women that actually took it as prescribed. 2 studies followed after that in post hoc analyses showed similar levels of protection, but these trials failed in the intent to treat, and as a result of this combination and relatively low percent in Caprisa, that was the end of it. I want to make an important point, though. We have taken a lot of lessons from this, but what's very different about vaginal formulation development is that in women, it is the epidemiologists tell us from pulling data from lots of studies that the risk of HIV transmission in women from receptive anal sex probably accounts for as much as 40% of HIV acquisition in women. And these products are vaginal and therefore, and do not reach the rectum, would not be safe in the rectum, as was later shown. So it's a very different landscape, much simpler in a way when these are studied in men that have sex with men, transgender women, because there's only one primary route of infection. So it gives us it gives us a chance when we get to phase 3 to have a much clearer picture about what a topical microbicide can do applied to a site of acquisition, to the rectal site of acquisition. So part of the failure here, as I just described, was all those blue boxes are all the questions that I just posed previously, and almost none of these were answered prior to launching into many of these studies. A lot of that work was done in parallel. Rectal microbicide development has been a driver of many of those studies and also a beneficiary from the whole vaginal product development. So we've been able to answer many of these questions. What I will say is I think, and I will put a plug in again, as Jim said, for the, I want to get this right, for the Choice Agenda webinar on the 24th of September, Do Vaginas Demand Perfection? And I can't wait to get to that webinar, because I think it is still arguable what is or are the anatomical target or targets for drug action, especially with regard to microbicide, but this is highly relevant as well for changing dose regimens of oral PrEP agents. And what is the drug concentration and duration at target? We kind of know what it is almost everywhere for many of the drugs, but we're not sure exactly which of those locations are most relevant for prevention. And I think this is especially true for microbicides, especially rectal microbicides, because we have no phase 3 data to support it. So the rectal program in many locations has answered most all of those questions. We're still working hard on the ones that I left highlighted in red. Now, I showed you the vaginal formulation studies in women. This vaginal formulation, as I mentioned, moved immediately over, well, as soon as we were ready for it. There was a phase 1 study done, RMP02, MTN 006. This was an NIH-funded study. In that very first study, that same vaginal formulation that was moderately, I would say, effective in women, was also a very high osmolality. That's that 3,111 milliosmoles per kilogram, high osmolality product. And because of that, it has good lubricating properties. Unfortunately, those lubricating properties and the whole hyperosmolality made it not well tolerated at all with rectal application. And also caused significant rectal toxicity, losing that very gentle single columnar epithelium lining the rectum and the colon. So that never went further into phase 2 studies. That osmolality was reduced, and a reduced glycerin formulation was studied in NTM 007 in phase 1, moved on to phase 2, and that's as far as that went. Only one prior rectal microbicide has been studied in phase 2. HPTN 106 that Mark will tell you about will be the second, and it is also a tenofovir-based product, but a completely different formulation. MTN 017, there were not significant safety signals in the study, but in careful analysis of the behavioral readouts, which is why this is so important, and Mark will point this out, applying this drug every day or even on demand with an applicator to inject the gel. This was not studied as a lube. It was studied as using an applicator and a gel. It will not go forward to phase 3 studies, but there were many things useful learned. The osmolality is further reduced in the rectal formulation. CHARM 1 and 2 studied this in phase 1. Unfortunately, at this point, there was great interest in looking at how this might work. This rectal specific formulation, how this might work as a lube. But when we looked at the data and a couple of other studies, it looked like to be able to dose this as a lube, to get over those dislikes of the applicators, this would have to be reformulated probably to 10 times the concentration. Now, that concentration has shown to be safe in lab animals, but this has not yet been studied. I think the future of an on-demand lube is challenging, but I think it's still something that should be considered. I think success with the tenofovir douche in HPTN 106 will be a reason to come back and look at this, because I hear questions about an on-demand lube more often than I even hear about an on-demand douche. And then finally, you'll hear all the details for the phase 2 HPTN 106. We've done 5 or 6 clinical studies already to prepare for this so that we can answer enough questions to have a thoughtful design, and we're ready to find out. This is a list of about, I guess, 10 or 11, 12 rectal microbicide products. They're all different in some way. Most of these are tenofovir, but there's 1, 2, 3, 4, 5, 6, 7 other drugs in these products. Of all these studies that have been done, these were all phase 1 studies, I think there are 4 that remain possibly viable, and for different reasons. The green arrow is the tenofovir douche that you'll hear from Mark about. The taf-elbictegravir fast-dissolving insert is at the bottom. That has completed a phase 1 study with rectal application. I think there's more phase 1 work to do, and then hopefully that moves quickly to phase 2. The tenofovir rectal formulation gel is the lube, which I said will be challenging, but I think we should reconsider this after we have success with the tenofovir douche, showing that the rectal microbicide can be effective, and then we consider even more formulations. And the Q-grifacin is another product that I think is promising. I think that's still in phase 1. I'm still waiting to see the completed phase 1 results, but I think that's still potentially in the game. So last comment, I started with vaginal microbicides. I'm going to come back to this. I think the optimal product would be able to be used both for on-demand use to prevent vaginal acquisition of HIV in women, and also for rectal sex in anybody that has anal sex. I made the point before that about 40% of HIV infection in women is calculated to be due to anal sex, not receptive vaginal sex. Douching, in studies that we've done in the US-Mexico border, is very common among female sex workers, but it is not really very well studied. We have far more information about rectal douching in people that have receptive anal sex rather than vaginal douching among female sex workers. Vaginal douching poses new product challenges and may well require a formulation change from the product we have currently. I expect that's probably going to be necessary, but I don't think it would change the still to be proven efficacy of the product. And we are doing vaginal douche optimization studies, both in terms of pharmaceutic development and also in terms of in vitro studies. So next step is REV UP, HPTN 106. And at this point, I'm going to turn it back to Jim to MC. And next up, you'll have Mark Marzinke.
Thank you so much, Craig. That was fantastic. What a great stage setting and context setting. Let's move right over to Mark, and then we'll see. We'll check in with folks if there are questions after Mark's talk before we move on to Marianne. Thank you so much.
Fantastic. And, thank you, Jim, for the invitation to present, and thank you for the wonderful primer, Craig. And, I want to welcome everyone, on behalf of our team. I want to thank you for registering for this webinar. And today I'll be discussing some of the foundational work that's been done in the field of rectal microbicides, building on, the IRMA initiatives built on what Craig has presented, as well as our current protocol, HPTN 106 REV UP. So I have no conflicts to disclose. And I think an overarching question that exists, why the need for a rectal microbicidal product? And I think I'm preaching to the choir, because for those registered for this webinar, the answer may seem apparent, right? We know that unprotected receptive anal intercourse, or RAI, is the highest risk sexual activity of HIV transmission. Additionally, we know that RAI is a very common human behavior. Humans of all genders and sexual identities practice RAI and enjoy RAI in both the developed and developing world. And lastly, we know that most people use products in preparation for or in conjunction with RAI. Therefore, from a sexual practice perspective, having a product that facilitates the delivery of a rectal microbicidal agent when someone needs it makes sense. We then have to think about the ideal characteristics of a rectal microbicidal product. And Craig did a very nice job of laying that out. So when we think about a target product profile, the initial focus tends to be on the chemical or physical properties of the product. And that may include its pH, its solubility, its viscosity, its ability to distribute into, for example, the colon. Additionally, there's a focus on the antiviral activity of the product, or how effective the microbicidal agent is, as well as understanding any safety signals or how the body processes the drug over time, which is known as pharmacokinetics. So these parameters can be evaluated using a number of experiments and models, including cell lines, as well as in animals. So while these are all really important parameters to consider, we can't forget about the essential role of end user preferences and acceptability in the product development pipeline. So as shown in this figure, there really is an interrelatedness between understanding the biology of the rectum and the mode of viral transmission, as well as developing products that are safe and effective, all while being acceptable and wanted by end users. As such, involving key interested parties in the product development pipeline is critical. So important considerations and rationale for pursuit of on-demand rectal microbicide products include the desire to have a product that is user-driven, right, and doesn't necessarily need a provider to administer. A product that results in lack of systemic exposure to a drug. Additionally, not all people want to commit to a drug or delivery modality that results in continuous or long-term exposure. Lastly, and as mentioned in the previous slide, efficacy is not the only product attribute that is important, right? Really the acceptability is a critical component of that. As Craig mentioned, from a biological perspective, there is a precedent for on-demand rectal microbicides, and we can look at current products and delivery systems that are on the market or going to be on the market for reference. So we know that single agent drug administration has proven effective in the oral, topical, and long-acting HIV prevention spaces. So we have experience with oral FTDF, the dapivirine intravaginal ring, as well as injectable cabotegravir, and more recently, lenacapavir. We also know that on-demand strategies have also proven efficacious. On-demand oral PrEP, or 211, has proven effective in persons assigned male at birth, and the tenofovir vaginal gel was also shown to be effective, as mentioned earlier. However, an important note is that topical products like the dapivirine intravaginal ring and the tenofovir gel were shown to be effective with high adherence. But the products that are currently available really indicate that a single drug can be effective for HIV prevention and that there is a precedent for products to be used on demand. So these characteristics have led us to pursue a tenofovir-containing rectal douche. So the tenofovir rectal douche can be used on demand so that it is utilized only when you need it, and it results in high local concentrations of the drug in the colon without resulting in long-term or systemic exposure. Lastly, and of great importance, it's behaviorally congruent. So within our group, we use that term behavioral congruence a lot, but what does that actually mean? So a product that is behaviorally congruent leverages an existing behavior, like douching before sex, to be used as a dosing strategy to reduce new behaviors required for product uptake. So if douching is a common practice before sex, and the rectal microbicidal product can be incorporated into one's normal douching practices, this can lead to greater uptake and more consistent product use. So behavioral congruence both relies on and aligns with existing behaviors. From a behavioral perspective, we have seen that having choice in terms of PrEP products is important, obviously a topic that's not new to many people registering for this webinar and members of our community. So this further motivates the pursuit of rectal on-demand products. So this work, which was published by Gordon Mansberg and Patrick Sullivan, summarizes survey data from 782 cisgender men who have sex with men from Atlanta, Detroit, and New York City. The data come from a baseline assessment of the mobile messaging for men, or M-cubed study. MSM not living with HIV were surveyed, and they were asked about their reported likelihood of using a long-acting HIV prevention modality, a sex event-driven pill, or an on-demand rectal product. The example on-demand products that were used in the survey were an anal gel or a rectal suppository. Participants also ranked their preferences for each type of product. Overall, most participants within the survey stated that they were likely to use each type of product when and if it became available. Overall, among participants who completed the survey, 67% reported likely use of sex event-driven pills, 74% for long-acting injectables, and 64% for on-demand rectal products. Based on the likelihood and preference assessments, actually two groups of potential product users were identified. Sexual event-based products, or more on-demand products, were more likely to be preferred by non-PrEP users, while non-sexual event-based products were more likely to be preferred by current PrEP users. So these data underscore the importance of choice, right? With some individuals preferring to use PrEP when they need it, as opposed to continuously. While there's merit in thinking about rectal microbicides in the abstract sense, there's also great value in understanding acceptability and use with actual products. So Jose Bauermeister led the MTN-035 study, which was also known as the DESIRE study. MTN-035 evaluated the safety, acceptability, and adherence of 3 rectal microbicide placebo formulations. So they looked at a rectal insert, a douche or enema, and a rectal suppository. In this study, none of these products contained a microbicidal agent or a drug. And the study enrolled 217 transgender men, transgender women, and cisgender men who have sex with men aged 18 to 35. And participants were randomized to different product sequences. All of them had experience or had the opportunity to use each of the products. And the products were used for 4 weeks each. They were instructed to use products between 30 minutes and 3 hours prior to RAI, following their usual practices. And this study was conducted in the United States, Malawi, Peru, Thailand, and South Africa. As shown in this table, acceptability and adherence to all products was high, with acceptability ranging from 66% to 73%, and self-reported adherence ranging from 74% to 83%. When evaluating specific preferences after product use, looking at criteria such as feeling clean after use, making sex more pleasurable, aligning with a participant's lifestyle, there was an overall preference for the douche, as shown by these dark teal bars. However, when looking at criteria such as serving as a lube alternative, the suppository was more preferred, as shown by the medium teal bars, and when thinking about discretion and ease of storage, the insert was preferred, as shown by the light teal bars. Within MTN 035, computer-assisted self-interview questionnaires were completed by study participants. As presented by Rebecca Giguere and colleagues earlier this year, the rectal douche was the most preferred product in the US, Peru, and South Africa. The suppository was preferred by participants from Malawi, and inserts were preferred from Thai participants. So these data highlight geographic differences in preference. In-depth interviews were also conducted in a subset of participants. And in those interviews, it was identified that preference for the douche was attributed to an overall familiarity with douching practices, that it was relatively easy to use, and it was behaviorally congruent. Also, it served as a twofer, right? It provided cleanliness and it had the potential to provide protection. So this formative work, in conjunction with the studies previously presented by Dr. Hendrix, further motivated us down the protocol development pipeline to pursue a rectal douche. So I'd like to now transition into discussing some of the work conducted by our group through the DREAM program project grant, which has led us to the REV UP study. And the REV UP study is a phase 2 crossover study of on-demand PrEP formulations comparing rectal and oral tenofovir-based PrEP, evaluating safety, acceptability, and pharmacokinetics and pharmacodynamics. The DREAM program project grant facilitated important work that evaluated multiple aspects of a tenofovir-containing rectal douche that were initially introduced by Craig in the previous presentation. And here we'll focus on the behavioral, safety, and pharmacologic aspects of that program and the product. So although we've shown data regarding the likelihood and acceptability of using on-demand products, we felt it was important to gather additional information on douching practices and community interest in a douche-based rectal microbicide. And this was achieved via international survey through the social media dating app Grindr. And this work was led by Rebecca and Alex Carballo-Dieguez. There were more than 5,000 respondents from across 52 countries. 80% of respondents from the US, 63% from Latin America, and 73% from Africa reported douching before sex. Further, more than 80% who douche do so less than an hour prior to RAI. More than 95% of participants across different geographic regions reported that they would be interested in using a medicated or microbicidal douche product. Further, among insertive partners, the majority supported their receptive partners using a drug-based or microbicidal douche. Respondents indicating a definite or probable likelihood of using a microbicidal rectal douche product are shown in gradations of blue. So the darker blue is definitely yes, the lighter blue is probably yes. In the left panel, these are among individuals who do not douche, and these are among respondents who do report douching. Rebecca and her team also reported on douching devices used across geographic regions. We know that under the umbrella of douching, different devices may be used. These may include an enema bottle, a rubber bulb, or a hose attached to a faucet, for example. So while the enema bottle and a rubber bulb were more frequently used by US respondents, as shown here, a hose was more common in Latin America, And the bulb or hose were more common from African respondents. So we can see here the hose from Latin American respondents and the hose or the bulb from African respondents. It was also noted that there was variability in the number of douches a person may use to feel clean. Among survey respondents, there was actually a pretty even distribution, with people using anywhere from 1 douche to 5 or more prior to RAI. And these are important considerations when thinking about generating a rectal product, particularly if there's an interest in using a product that aligns with existing practice. So as we have to start somewhere, and a topic that my colleagues will address in the Q&A. Within the REV UP study, we will be evaluating the addition of tenofovir, that is in a sachet, shown here, into an animal model. And while there's future interest in evaluating different douching devices, within the study, we're focused on drug administration via the bottle. We have evaluated the tenofovir douche in 4 phase 1 trials to date. A total of 42 participants have been enrolled, with 110 total doses being administered to date. A summary of these studies are shown in this table. And this slide really focuses on the safety observations when using the tenofovir douche. We did not notice any changes to rectal tissue after using the tenofovir rectal product, and that was based on collecting biopsies and looking at histologic analysis. Across our phase 1 studies, there were 5 grade 1 adverse events that were attributable to the rectal douche. There were 2 instances of pelvic discomfort, 1 of rectal dryness, 1 instance of blood-tinged mucus in a patient with a history of inflammatory bowel disease, and 1 instance of bloating. Overall, based on among these 42 participants and 110 total doses administered, the safety profile for the tenofovir douche looks pretty good based on these findings. Okay, so now that we have an understanding of the behavioral and safety components of a tenofovir containing rectal douche, we also want to understand how the drug distributes, particularly within target rectal cells. And to go back to Dr. Hendrix's earlier point, where is the anatomic site? What is the target of interest? For the purpose of these analyses, really what we're looking at is the distribution and activation of drug within rectal mucosal mononuclear cells. And we're specifically interested in measuring the active anabolite of tenofovir, which is a molecule called tenofovir diphosphate. So tenofovir diphosphate is the form of tenofovir that within a cell can inhibit viral replication. So we were therefore interested in measuring tenofovir diphosphate concentrations in rectal mucosal cells at various time points after the douche was administered. Additionally, since we know that individuals may use more than 1 douche to feel clean, we also wanted to evaluate the sequence of douching on drug concentrations. The good news is, as shown in this graph, rectal drug concentrations are higher with rectal application of tenofovir as compared to oral on-demand FTDF. So for reference, the purple and pink lines are showing rectal tissue concentrations over time with oral on-demand FTDF. The solid lime green line are showing rectal cell concentrations after administration of a single tenofovir douche. And the solid blue line is showing rectal concentrations after 3 tenofovir douches are administered. However, the use of tap water douches before or after a tenofovir containing douche does lower drug concentrations. So we can see here that the solid forest green line represents the tenofovir diphosphate concentrations in target cells after administration of a tenofovir douche followed by 2 tap water douches. And the olive line represents the administration of 2 tap water douches followed by a tenofovir containing douche. We also simulated rectal tenofovir diphosphate concentrations when tenofovir was given at the beginning and end of a douche sequence. And this is shown in a dashed teal line. And what we can see is, that basically mimics what you see between the tenofovir tap water and a single tenofovir douche. So based on these observations, and to ensure enough tenofovir penetration occurs into the rectal compartment and our target cells, tenofovir douches will be given in this trial at the beginning and end of a douche sequence. So now that we know that rectal tenofovir diphosphate concentrations are higher after douche administration, we also wanted to know if the concentrations in rectal cells could inhibit replication of the HIV virus. So using biopsies that we collected from study participants, we used an experimental model in which biopsies could be exposed to the HIV virus, and we can determine if drug concentrations within the tissue could inhibit viral replication. And what we showed was that there was less viral replication after douche administration as compared to oral tenofovir dosing. So on the y-axis here, we have our marker for viral replication on a logarithmic scale. We're looking at P24 antigen production. And on our x-axis, we have rectal tissue tenofovir diphosphate concentrations, which I described on the previous slide. Looking at this graph, and it can be hard to interpret, really where you want to be is in this lower right portion, where you have high local drug concentrations and very little viral replication. And what we can see here is that there's a cluster of blue circles. And these blue circles are from biopsies that have been challenged after exposure to the tenofovir douche product. And specifically, this is within 1 day of tenofovir douche administration. For reference, the purple triangles are for individuals on daily oral PrEP. So what we can see here is that, obviously these purple triangles have lower concentrations and have greater viral replication as compared to the tenofovir douche. So this is helping assess our pharmacodynamic output, right? Are the concentrations that are found in these cells effective in inhibiting viral replication? It's also important for us to understand how the sequence of douching relative to sex could impact the distribution of either the drug or the virus. So we actually did this experiment by using labeled molecules to serve as surrogates of HIV and tenofovir. 5 participants were included in this analysis, and we evaluated distribution of the viral or drug surrogates if douching occurred before or after a simulated RAI. And using image analysis, we looked at our tenofovir surrogate, which is shown in orange, and our HIV surrogate, which is shown in this bluish purple. As you can see, when the douche is administered before RAI, we see distribution of tenofovir or the tenofovir surrogate throughout the colon. But we don't see distribution or migration of the HIV surrogate. However, when douching follows RAI, not only do we see, migration of the tenofovir surrogate throughout the sigmoid colon, we also see migration of the HIV surrogate. So based on these findings and the desire to not further distribute viral particles throughout the colon, it's recommended that the tenofovir douche be administered before RAI. So now that I've covered the road to REV UP, or HPTN 106, I want to take a few moments to discuss what we will be doing in the study. And as mentioned at the end of this presentation, I'll transition to my colleague, Marianne Gilday from FHI 360, who will provide some additional logistic details about the protocol. So within REV UP, our primary objectives are to describe the safety of the on-demand rectal tenofovir douche and compare it to the safety of on-demand oral FTDF. And we want to also describe the acceptability of these products, describe and compare the acceptability of these products. Our secondary objectives are to compare participant preferences between the on-demand oral FTDF tablet and on-demand rectal tenofovir douche after products are used. We also want to describe adherence to both products and as well as describe the systemic and local drug concentrations of both products. The overall purpose of the study, as indicated in the title, is to establish the extended safety and acceptability of the tenofovir rectal douche. This will help advance it down the product pipeline for an on-demand rectal microbicide. This is an open-label comparative randomized trial. We will be enrolling persons assigned male at birth who are 18 years of age or older, not living with HIV, and have a history of RAI of at least 5 times in their lifetime and once in the last 3 months. We're also looking for individuals who douche regularly before RAI. Our target enrollment is 150 participants across 8 US sites, and Marianne will expand on that a little bit later. A picture is worth 1,000 words, so our amazing team at FHI have actually crafted this image, which we've used multiple times to summarize our study design. So participants will be randomized 1-to-1 to an 8-week sequence of using oral 211, which is shown here, or the rectal tenofovir douche. This is followed by a 2 to 4 week washout, and then alternating to the other sequence or the other regimen. So we can see here that participants will be on study for approximately 19 weeks. The study products that will be used will be tablets containing emtricitabine and tenofovir disoproxil fumarate, also known as FTDF or Truvada. These will be administered to participants during the oral 211 sequence. The other product will be a tenofovir powder, which contains 660 milligrams of tenofovir. As you can see, these are provided in sachets that will be poured into enema bottles, which will then be filled with tap water and gently shaken before use. The tenofovir sachet is perforated to facilitate easy tear off. It also has a small opening to control transfer of materials to the enema bottle, and the size of the granules allow for easier pouring. Additionally, the sachet is actually pretty small to facilitate portability and discretion. Although the primary objectives of the REV UP protocol are to evaluate safety and acceptability, we'll also be looking at drug-based objectives within a nested pharmacokinetic pharmacodynamic substudy. The substudy will enroll up to 28 individuals, and this will augment existing data from our DREAM phase 1 program. Chiefly, we really want to understand rectal and blood-based drug concentrations between the 2 regimens, as well as evaluate how those concentrations in rectal cells can inhibit viral replication in our tissue explant model. We also aim to look at changes in rectal tissue histology after each product use, and to look for changes in the rectal microbiome over time. These assessments are going to be really helpful in informing the target product profile and will allow us to more comprehensively understand the pharmacology of not just the tenofovir rectal douche, but also on-demand oral FTDF, where several of these assessments have not been done before. So in summary, the development of rectal microbicides can facilitate expanded choice in PrEP options, may be used in a way that is acceptable to end users and provide protection when it's needed. HPTN 106 REV UP will evaluate the safety, acceptability, user preferences, and pharmacology of a tenofovir douche as compared to on-demand oral FTDF. And we think, and we hope that you agree, that this study is critical in the product development pipeline for an on-demand rectal product. I'd like to now turn it over to Marianne to discuss some of the additional logistics around the protocol.
Thank you so much, Mark. And before we turn it over to Marianne, feel free to get yourself ready. There is a clarifying question in the chat, and we will take a moment if people want to ask a question verbally. You can raise your hand. But Mark, for you, this is from Matt Rose. What prevention strategies and support will be provided for participants during the washout period, as 2 to 4 weeks can be a long time for people at high risk?
Yeah, so I think that's a great question. And again, this is obviously a study in which there's going to be continued interaction between study coordinators and the participants, including during that washout period. The reason why we need that washout period is based on our analytical capacity to detect tenofovir after last product use. So we think that the 2-week period is enough to wash out any tenofovir that may be hanging around in anatomic sites. But from a logistic perspective, building in some wiggle room around that, we think is helpful. Again, I think continued communication between the participant and the site will be critical. And I will turn it over both to Marianne and Craig if they have additional thoughts on that.
Marianne and Craig, do you want to jump in on that? Or we also have another question as well.
Yeah, so I think the other reason for the washout, because we have this for everybody in the main study and the sub-study, is that we wanted kind of a behavioral washout as well. So I think we had made this as short as possible, but still be able to distinguish the two periods and the two different products from each other. The way the inclusion criteria are written, we are not looking primarily for a very high risk population. We're looking for I would say, moderate to low risk population, which was a requirement really from it was a requirement with the FDA in prior studies. It was desired by DAIDS, by our funders, and I think it's an appropriate approach for something that remains unproven. We have counseling, we have condoms, we have lube, we have as many kind of supports as we can provide during that interim. And in addition, there are relationships that will have been built up with the study team over an 8-week period. So we're hoping that is adequate, and it's also a point that's reinforced, Matt, that this is very important. And while the on-demand oral is proven effective, the douche is not proven effective, and this is consistently messaged in recruitment, in consenting, in the ongoing counseling that goes on through the active periods and the washout in the middle. So I hope we have enough for this, and we will all be highly available to the study participants. And we share your concern. So we've tried to be very thorough in addressing this. I don't know, Marianne, if you want to add anything.
Yeah, I think that about covers it, including that we have specifically called this not a medicated douche yet. And so the language around that has been very clear in the participant-facing materials that this is still under study. So to continue with the condoms throughout the study, including the washout.
Thank you all. And so one more clarifying question, and then we'll move on to Marianne, but this is from Julie Patterson. If people are on a current TAF-based PrEP, or people who are current TAF-based PrEP users, can they enroll in this trial? Would there be a washout? If so.
Yeah, so that's a great question. So for individuals who are currently on PrEP, part of the inclusion criteria is for there to be a washout before being enrolled in the trial.
Excellent. So thank you for all of that. Why don't we go ahead and segue over to Marianne, and then we'll bring everyone back for lots of questions and comments for the rest of our time today. Go ahead, Marianne.
Okay, Jim, can you see the presentation slides?
Yeah, it looks gorgeous.
Okay, thanks. Okay, good morning, good afternoon, good evening, everyone. Thank you so much for being here. Obviously, we're very excited about this study. I'm Marianne Gilday, and I am a clinical research manager at the HIV Prevention Trials Network. So I oversee the operations of the REV UP study, otherwise known as HPTN 106. You'll hear those sort of interchangeable. You've heard from Craig Hendrix and Mark Marzinke about the background of rectal microbicides and how we got here, and I'll talk a little bit about study timelines and what's next with the sites. So first, who are we? The HPTN Leadership and Operations Center is located in Durham, North Carolina at FHI 360. We have a team of people overseeing the clinical operations, communications, community engagement, finances, contracting, network governance. Pictured here are some of my amazing colleagues from our recent meeting in DC, many of whom work on this study, many work on other HPTN studies. So our other HPTN partners are the Statistical and Data Management Center located in Seattle, Washington, and the Laboratory Center at Johns Hopkins in Baltimore, Maryland. And for the REV UP study, we have a behavioral team at Florida State University. So you can see that there are teams of people working on the entire study from a great level of oversight. For the audience today, I want to just highlight some of the activities we've done regarding participant-facing initiatives and those things that we have accomplished. I mean, I can write a really great protocol and the finance team can really balance a budget, but I'm not sure that's what you want to hear about today. So I'll show you a little bit about how we've engaged the community so far and then get into the sites and the timelines. So as we wrote the protocol, the LOC community engagement team worked closely with local sites to solicit feedback throughout the protocol development. So for example, the medical world can get extremely technical when it comes to rectal anal intercourse and douching, but certain wording just does not translate to real world participants, particularly when reading an informed consent. So based on feedback, we would adjust the language and make it more relatable, more real world for what the participant would need and still conveying medical aspects. The community engagement team also received feedback on participant materials and questionnaires. Again, making sure the information is clearly conveyed and appropriate for the audience. And once the study launches, the team will disseminate educational materials and host webinars to continually engage the larger community. We want continual feedback, so they've also assembled an ongoing community working group to have them bring their questions to us and be able to really give us feedback on how it's going and what maybe needs to be changed. So we also have a communications team here. They work with the content experts, the medical team, on creating materials for the participants. There are detailed scenarios that could be helpful to the participant on how to take the oral on demand and how to use the rectal douche. Some of these scenarios depend upon how much sex a person is or is not having in a given week. So the team created scenarios based on some anticipated situations that a participant may find themselves in. And the communications team also, along with the pharmaceutical project manager, seen here, also created a video on how to prepare the rectal douche that the participant will be able to watch. And finally, we have an amazing graphic designer who created the REV UP logo, where you see the enema bottle and the pills peeking through, and along with that study schema that Mark showed earlier. Okay, so that's the big picture. Who's actually in the weeds with the participants? These are the sites that will be enrolling the 106 study. So we have a bit of East Coast representation. We have a little bit in the Southeast and one on the West Coast. So the University of Alabama at Birmingham is in red. The University of North Carolina, Chapel Hill is in yellow. Fenway in Boston, gray. The Hope Clinic in Atlanta is green. Johns Hopkins in Baltimore, light blue. UCLA in Los Angeles is that teal color. University of Pittsburgh in dark blue, and Cornell in New York City is in the purple. So each site has, you'll hear the term teams a lot. Each site has a team of people working with the principal investigator to make the study happen at the site. So the PI may be listed here, but there are many, many people at each site. For instance, at a minimum, a clinical research coordinator, a lab team, a data manager, a research pharmacist, community engagement team members. I mean, the list goes on. So we work closely with all of them, making sure that each entity is doing the study correctly. The other thing that the Operations Center does is send the supplies to these sites for the eventual distribution to the participants. So each site will start with a partial shipment that will just take up a little less space because it can be a little bulky. For example, each site will receive 75 enema bottles and 300 enema tips to create what would be the full enema bottle for the participant. Additionally, the sites receive 65 at this point in the small shipment, 65 large 4-ounce tubes of lube, that's on the left, and more than 600 smaller, flatter, portable lube packets seen on the right in the other image. We also have discrete bags, no logo, no HPTN, no REV UP on the bags. Each site right now will get about 15 plain bags that a participant can receive, put their supplies in, and have some discretion around it. Okay, so we've been here for more than an hour, and we've presented today on the background, how we got here, why this modality, the reason, and there's been a lot of buildup and a lot of revving up, so to speak. But when will this study open? So here are a few key timelines. I mean, this protocol writing has gone on for a couple of years, but I will just give you the past few months. The Division of AIDS approved the protocol in late April, and the Operations Center received funding. The FDA cleared us and said we were safe to proceed in May. Site training started in June. We hosted both in-person and webinar trainings, because as you can hear, there's a lot of elements to this study, and each element needed its own training. The 8 sites have been working on site activation, which can be up to several months due to the regulatory aspects of an IND study, the contracting, the lab activation, training a counselor, receiving those supplies and the study product. But happy to report that we will have a few sites up and running and open in September. Okay, so you've heard this. Maybe you're interested in learning more. Maybe you would like to see if you or someone you know is an eligible candidate to enroll. There are a couple places that you can get more information. We are listed on clinicaltrials.gov, the National Registry of Clinical Trials. There's a distinctive number listed there where you can find us. It gives an overview of the study and who to contact at one of those 8 sites. The sponsor is NIAID, and there's also that link there that I think Jim dropped in the chat. Additionally, this QR code and website will take you to the HPTN 106 website and provide you with the contact information at each of those 8 sites. They would be your best resource for screening. And you can also like and share and follow us on all the socials at HIV PTN. Finally, this is an overall promotion for the Red Ribbon Registry. This is a database of people who want to support HIV clinical research by participating in studies. The registry allows researchers to quickly find people who might be a good fit for a study based on age, location, health factors, and other criteria. So some of the REV UP sites are also registered there as well. Finally, we'd like to thank our funders and all of you for being here today and attending and listening. And it sounds like you guys are as excited about this study as we are. So appreciate your time. And I think there'll be some more time here for questions. Thank you.
Beautiful. Thank you so much, Marianne. I'm going to ask Mark and Craig to show us your beautiful faces once more. We get you spotlit here. And I see your hand raised. Matt, we'll get to you in just one hot second. So I just don't want to lose track. There's a couple of questions in the chat, and I think the last, you might see it. What is the lubrication that you all are recommending participants use during the study?
So we are recommending and providing a water-based lubricant, Good Clean Love, and the primary reason for that is that there's an awful lot of study that have been done of water-based, and especially water-based at different osmolalities, and this is a relatively low osmolality that is proven to be safe to a rectal mucosa. linings. So we are making it easy by providing the lube throughout the study as needed, which we have done in many studies. There may well be other products that are appropriate, but we're just trying to do this somewhat for consistency and ease of access to that. The oil, I would say, is not a good idea for a number of reasons. Primarily, it may well be highly destructive of the very delicate lining of the rectum and colon, and very likely accelerate, or so I should say, increase risk. I'm not aware of as much data on silicone-based products. So the reason we're not recommending that and not providing that is the lack of data. So we're going with what we have the most experience with and the best data that goes directly to safety and rectal health.
Thank you. And I just want to touch on this a little bit more. Jeannie Barron from AVAX Comms team was on earlier and asked about osmolality and what that actually means. And Mark gave a definition, but can one of you just describe quickly why osmolality matters and why all the kind of work that happened around lubes, we started to look into the osmolality of different lubes and which is, quote, safer or not. There was a fair amount of research that happened in that space. Charlene Dezzutti, which many people remember, God rest her soul, did some really great work in this area. So can you just give a quick description of why osmolality matters and what we should be looking for in terms of products?
Yeah. So Mark gave a great description of what osmolality itself means, and historically lubes have worked by being high osmolality, because that high osmolality in the rectum draws fluid from the tissues very rapidly. If you have a product that's isoosmolar, then there's no rapid fluid flux and no stress on the cells themselves. If you have products that are hypoosmolar, we expected there might be something going on, but we found them all to be very safe and no damage with hypoosmolar, including tap water. We've done studies with a wide range of osmolalities of lubricants. Ed Fuchs published this paper, I think, in JID in 2007. It was one of the first things we did, and Ed was involved with most of the studies that you'll see in the reference list. He's just been a tremendous colleague, and now he's enjoying his retirement. But we've showed that there's very few over-the-counter lubes that are in the iso or hypoosmolar category. Good Clean Love is one of them, which is why we use it. So the douche is also same kind of issue. If we used a hyperosmolar douche like the traditional Fleet enema, that is very hyperosmolar. That's how lubes work. That's how douches work. They draw massive amounts of fluid very rapidly into the lumen, and that's destructive of that very delicate single columnar epithelium. You lose the mucus, you lose the cells, and then you have ready access of virus directly to CD4 cells that are sort of circulating, in and around the surface anyway. But that removes an important mucus and cell layer that we and others have demonstrated is quite toxic. That's what you see when you use that hyperosmolar vaginal formulation. Vagina is much more tolerant of those kinds of stresses, so not the same kind of a problem, although it does cause some symptomatic issues because there's lots of fluid, and the women in some of the early studies frequently complained of wet panties and lots of unwanted drainage from after application, soon after application, for the same reason. So hopefully that kind of explains the reason for both lubes and enemas that we want to use either iso or lower. We use the much lower hypoosmolar formulation because it greatly accelerates movement of drug to the tissue surface for rapid absorption. Tenofovir, the formulation of tenofovir, the form of tenofovir, the chemical structure we're using, is not very well absorbed. If it were taken orally, very little would get in. But when we use this hypoosmolar as an accelerant of sorts, I mean, kind of with a different purpose than the way that's normally used, but to really speed up very rapidly. So we get tenofovir into tissue quickly, and then the phosphorylation can begin very rapidly. And Mark nicely showed how rapid we get very high concentrations compared to oral dosing. especially if they're both dosed at the same time. So we've looked at these comparisons directly. So that was all part of the design. So thanks, Jim.
Thank you. And Matt, I appreciate your patience. I want to just pull up one more question from the chat because it's about lube again, and we'll try to stay on topic here. So Ben is asking about, analysis, and if someone was to use their own favorite lube, like silicone lube, during the study, how would that potentially impact your analysis if they're not using the lube you provide?
It may add a little bit of heterogeneity. What the biggest thing I would worry about, based on what I just said, is if someone is going to be using an oil-based lube and that increases the risk of HIV acquisition, we are certainly tracking HIV throughout the study. That's in a way that's always our biggest concern. That's the whole reason for the product, but especially as it's unproven, we're going to be looking very carefully at that. Hence the discussion. Great question from Matt earlier on this topic about the level of risk. The things we're measuring are not to be very sensitive to that. So what I would suggest is that and Mark pointed this out, all this adverse effects we've reported, because we write down everything that is verbally reported to us. All of those are the same kinds of side effects, adverse effects that people mention when they douche. There's nothing different in the spectrum of things that we've seen than usual douching. I expect that if someone uses their regular lube, it's going to be all the usual, if there's ever any complaint, that might be the usual complaint. And I think that would be part of the usual back and forth if they're reporting adverse events, and the study coordinators are going to be addressing that and digging a little deeper to try to understand if there's a new symptom, what's new. Maybe it is the lube we're giving. Maybe it's the lube they've always been using and are more familiar with. And of course, the participants will have the best knowledge of their own history with that. So it'll be our responsibility to dig into that, to find out what's going on, and then we'll look for patterns in that. But I'm sure people are gonna do that, and I have, other than the caveats I gave, warnings. I mean, I would raise it to the level of a warning for certain types of products. As long as they're using a water-based product, especially if it's an isoosmolar, I don't think it'll have a big influence. The volume of the lube that actually gets in is relatively of a hypoosmolar lube is so small relative to the douching that they're going to do that I don't expect it's going to change much of anything in terms of the rectal lining. In fact, the lube has a very limited distribution compared to where the douche is going. We won't be doing any assessments with biopsies in everybody in the main study to see if there's any toxicity to study lube or non-study lube. We're using the study lube because we know that's not toxic. So people can do that. We're trying to make it easier to do what we're strongly suggesting by distributing that, and hopefully the dialogue, if people are using something other than that may inform, we're going to track that. We'll try to learn the best we can from that. And clinical research isn't always clean and unmessy, and that's where you learn stuff. So we're very open to that.
Thank you. All right, Matt, you have been so patient sitting there in your gorgeous office.
I mean, I'm getting a second
background.
question in, so I'm just happy I get the second question in.
There you go.
I'm looking at the number of enrollees and thinking about the context of where we're doing this study. And I noticed I didn't see or hear a mention of what you're doing in regards of race. And even though we're not looking at a phase 3, we know that socializing products within communities is very important. And where we are at with the epidemic in the United States would mean that you would primarily want Black and brown folks in the study. Do you have plans on how to reach those populations so that at least they're getting socialized to the concept of this being a prevention option for them, and potentially bridging you to a third phase study if the data shows that we're headed there?
Thank you, Matt.
Is Jonathan on? Does Jonathan want to address that? Because I think that may be the best. A great question, Matt. Thanks as always for having the good questions. Is Jonathan with us, Marianne?
I am on. Can you hear me?
Okay, great. Yep.
So it's a really good observation, Matt. What we aspired to do when we selected sites was make sure that we got a good sample of sites, sort of, it's got access to diverse populations, because we want to make sure that we're hitting as many of those folks, you know, people of color, transgender women, as we possibly can. I'm recognizing that it's not a phase 3. We only have 150 slots. We did want to be intentional about reaching those folks, given the HIV epidemic amongst those communities.
Thank you very much for that, Jonathan. And thinking about other communities, and I think this would be, you know, if hopefully we're able to move this work forward. But Angelo from AVAC, who is no longer on the call, had to drop off, but he had asked before he left, what are plans for including people who can get pregnant? So, and cisgender women, people who might be pregnant and lactating. And we heard quite interesting data about, you know, the relevance of unprotected receptive anal intercourse among cisgender women, people with vaginas, people who can get pregnant, and wondering about what the pathway looks like for including them in the future. And feel free to jump in.
I guess we're here to answer the hard questions.
That's a hard question.
You know, some of that, two parts. So for folks that are capable of being pregnant, we want to get to that population as soon as possible. The question is how fast we should do that. And I think we should be taking the lessons from vaginal product development, which I think has moved too slowly, but with appropriate caution, to open welcome enrollments and not dealing with it as pathology, but dealing with potential for pregnancy as a celebration, but something that raises additional issues. We are focusing here, and the other populations that I think you mentioned transmasculine. So the concern there was raised, and actually we had originally included this for the study population, to have trans men in the study, and the community on several occasions provided feedback that they were concerned that the duration of the study with 2 unproven strategies, and I say 2 unproven because the oral 211, which is the control, which is what's going to provide some level of protection for 8 of the weeks in the study, has not been proven in that population. I think that may be a topic that comes up in the September Do Vaginas Demand Perfection webinar. So I think that's a very complicated issue. Community raised those concerns. We took that very seriously and removed the trans male population from that. So the other variable that's hard to get at, and we wanted to here we're interested, I will say, in rapidly accruing in a population where we found it to be safe. We've been limiting it largely for the purpose of looking just at individuals that have receptive anal intercourse as a risk, and the attendant behaviors that go along with that as the primary population. The question is now, as we are beginning discussions about design of phase 3, there's not much that's different that we should treat differently anatomically in women that could be in individuals that might have a vagina and along with their rectum, as everybody that will be enrolled will have. So that's going to be part of the next discussion for the phase 3. The things that are limiting for that are not nearly as much data on receptive anal sex practices, parasexual behaviors like uses of lubes and douches in women that have receptive anal sex and may also have vaginal sex, other than the idea that the risk from receptive anal sex is really quite high. And we actually led with that. And we're very interested, and that's why we're working on a vaginal formulation that we hope can be used both vaginally and rectally. One cannot protect the other. That remains, and if I didn't say this, I should say this, that is perhaps the largest single limitation of a topical microbicide in women. This is not true for an individual that only, I should say, that only has, that does not have a vagina and whose primary risk is receptive anal sex. So it would apply to anyone whose only risk is receptive anal sex. And that is the cleanest population. And by clean, what I mean is easiest to get a statistically clear, unquestioned answer. And you saw all the issues that occurred, understanding what's the background of anal sex with the topical vaginal program. That is kind of muddied the waters to understand how well do these things really work. Are they only at 75 and 85% because there's enough receptive anal sex going on, even at a low frequency, but at very high risk compared to vaginal. So this is going to be central to the discussions because we'd like to do it. I would say we're going to start with the assumption that we'll do that. The question is going to be what numbers, what kind of durations, what are other caveats, how we'll manage women that might become pregnant during that study, et cetera. So you can hear me struggling with this. We all need to be struggling with this. And I think we should begin by putting it on the plate as included. And then if there are very strong scientific and statistical rationale, we'll consider how to mitigate those concerns if possible, or limit in the enrolled populations.
Yeah, and maybe just to augment that briefly, so I was looking in the Q&A and to reaffirm what Craig mentioned, this study is enrolling people assigned male at birth, right? So transmasculine individuals would not be eligible for the study. And, like really part of our initial ideation and conversations around including either transmasculine individuals or cisgender women and people assigned female at birth was also sort of the unknown landscape of on-demand oral PrEP in those populations. And I think your upcoming webinar on September 24th will help get to that. And I think regarding, some of the formative work that's been occurring through, the long-acting agendas amongst pregnant and lactating individuals, really leaning on some of the guidance and recommendations that have come out of those trials, as well as the WHO's pregnancy toolkit, are going to be important considerations when looking through the lens of a rectal microbicide. And that would be obviously, extended safety during pregnancy, as well as in the postpartum period, as well as additional pharmacologic analyses, which in the landscape of an on-demand agent may be harder to characterize. But I think those are important questions to consider, and we should obviously craft those in collaboration with other key interested parties.
Thank you, thank you. So we're getting towards the end, but we had a ton of great questions come in during the registration process, and I want to just see if we could tick off a few of those with quick answers, if possible, before we close out. And we may go a few minutes past the time. I hope that's okay. So I've seen this myself, and I'm sure you all get this question a lot. In terms of how this douche works in terms of preventing HIV, is it just effective for the bottom partner, or could there be impacts for the top in terms of effectiveness? And what about any kind of side effects that might happen in terms of the top, the insertive partner?
Yeah, so I think that's a really good question. The douche is designed to protect the bottom. I think studies of the top remain in the future. We know that there's been some previous work, for example, with dapivirine gel that has been applied to the penis, but at least to my knowledge, there's been no testing for any product when administered only to the receptive partner. So, Craig, do you have any additional thoughts on that?
Yeah, I would only add to that this will be the first time that this product is used kind of, as desired out in the community with the usual partnerships. We've only been giving single or multiple, but rapidly numbers of douches in the clinic. And we've never been able to send the product out before. In phase 1, we didn't think it was safe to do so. Now that we've established safety, at least for the rectum, this is the sort of data that we, again, will be open to capturing if there are complaints or feedback. Again, this will be the first that we get that. So we're quite interested in that. It was not a complaint in the prior tenofovir gel studies, which were sort of the ones that were studied most. That was in the prior MTN-017 study that had daily and on-demand use of the tenofovir gel.
Thank you. And I know Mark touched on this, but I think it's worth just emphasizing because it's confusing. As we know, the way people douche in very different ways, and some people may need one douche to have the hygiene they want, and other people may use a douche multiple times till, quote, the water runs clean until they have the hygiene and the freshness they want to have. So in terms of dosing this rectal douche, how does that work? Can people still, do their 5 bottles to get the cleanliness they want and then do the medicated douche? Can you just re-emphasize like how that works for people who may have different needs in terms of having that water run clean?
Yeah, I think it really goes back to this idea of behavioral congruence, right? We want individuals to use the tenofovir douche, but we obviously also want that to align with their practices. So what we have put in the protocol, and is going to be in a lot of our participant facing materials, and in consultation between study sites and the participants, is really to kind of where we've leaned into a bookend approach, right? So that the first douche that you use should be a tenofovir-based douche. And if you feel the need for additional douches, then you should, using your standard methodology, perform additional tap water douches. And I think, Jim, using your analogy, when the water runs clean and you feel like you're done, you do one more tenofovir douche, right? So it's kind of a book-ended approach. And that goes back to some of the pharmacologic analyses that we've shown, because we know that the use of tap water douches either before or after a tenofovir douche can result in some drug loss. So this book-ending approach, at least based on our simulations, is really meant to help maximize penetration of the microbicidal agent, right, the antiviral agent into the rectum.
Excellent, excellent. And now I did say try to answer these questions quickly. I am going to give you a question that's it's definitely not a quick answer, but give your kind of top line thoughts, because this could be a whole webinar on its own. But in the context of multiple now diverse and effective PrEP options, and the landscape that we could likely in the next year have yet another long-acting injectable to join cabotegravir. Lenacapavir is probably coming next. What would a phase 3 clinical trial for a rectal microbicide douche look like in that context? And I know, like I see the smiles, this is not a 5-minute answer. This is a 105-minute answer, but can you give us, your sort of immediate thoughts about how you would address that? And maybe speak to also this notion of efficacy. And the field is really driven by efficacy, and efficacy equals the best thing, right? Or a lot of people think that. But in terms of how people use contraception, it's not always about the most efficacious product. It's the one that they want to use, and then therefore the one they want to use is the best product for them, whether or not it's 100% efficacious, right? So what are you all thinking about for that phase 3 in this really challenging and exciting dynamic space that we are in the HIV prevention world?
Am I going to do this one? So, Jim, this is what I call the $250 million question.
Right, in the last 2 minutes.
Right. So there's 2 parts of that. One, I would say we now have 2 very different strategies for the design and statistical analysis of phase 3 randomized controlled PrEP trials. And I would say that the lenacapavir study, we have, that is such a novel design in a way that once we can see all the details, that really is going to be a place to start the discussion to see if there's something like that we would do, or something that is more traditional. I think the concerns are how to get that 250 million down to a smaller number. And we don't need to be as complex as that. But I could even imagine, because of the absence of sufficient data to either present to FDA or for FDA to agree to approving a 2-1-1 regimen, which is our best comparator. And it's why we've been very deliberate in choosing folks that douche and are interested in on-demand, so that the oral 2-1-1 with FTDF is the appropriate comparator. And that's why we've got this crossover design where everybody gets each, and in a paired analysis. The challenge is that neither of those have been proven and then presented to the FDA for approval. So for FDA to approve a design, will they require an oral daily FTDF as a proven acceptable product with daily use, and also have some randomized on-demand and some to on-demand douche? That gets very complex, but it is 3 arms. We've seen 3-arm studies. I like 3-arm studies because you can learn a lot more, and it's highly efficient in that sense. So that, I think, is a starting place for how to do this. Questions about recency assays and how you build in controls to understand that, very provocative. And I think that's where looking carefully at, under the hood with actual numbers now from the purpose study, plural, eventually, that we'll have to look at. And those discussions should begin right away. The other point you were making is, does something have to be 95 to 99% effective for it to be approved? And I don't think that's true. I don't think it does. I think that is not necessary, because, and I think the vaginal microbicide studies have made this point, that in some populations, and we're talking about tens or hundreds of millions of populations, that a ring it may have a little bit lower efficacy, but because the uptake and persistence and adherence may be superior in some geographic locations to an oral daily pill, and an injection may or may not be available, the ring becomes the best option for many of those women. We are selecting our population carefully for those that we think that those are designed for. The majority of people probably are not interested, and I cheer them on that they have so many options for what works for them. We're working on a smaller piece of this, and whether it's a third or a half, I don't know. It's, the surveys that Mark showed you suggested maybe north of half, but it's certainly a significant chunk. This option, we hope, will be available to individuals to make that decision. I hope that there isn't an absolute standard threshold that we have to exceed, because of all the things we've learned that I think have been the right lessons from the vaginal product development. That systemic isn't for everybody. A douche is not going to be for everybody. Topical, on demand may not be for everybody. But it is, I think, for some people. We want to put it in play so that people can make their own individual decisions at what time which product best fits into their active lives.
Beautiful. Thank you for that, Craig. And I'm gonna, in our last moments here, I'm gonna offer, if Marianne or Mark want to send people away with one nugget that you remember and take forward after this webinar, what would that be?
Yeah, I think the nugget is that in order to push a product forward, it takes collaboration across the spectrum. And I think that, Jim, you inviting us here, I think wonderfully illustrates that. And, I think that this protocol may seem long overdue, but it really is built on decades of advocacy and research. And, we think that this is the door to that $250 million question.
Thank you, Mark. And Marianne, do you have a nugget?
I would just say if this interests you and your friends and colleagues and want to look into participating or spreading the word, we would love to have you engage with our sites who are top-notch in this field, and we also welcome other feedback along the way. So we have adjusted this protocol a lot. I can probably say for sure it will be adjusted again once we start enrolling, and continuously receiving feedback on it is always important. But thank you, Jim.
Thank you, Marianne, and I want to thank the HPTN for partnering with the Choice Agenda on this webinar. It was great collaboration. Let's give some love to our amazing presenters, Craig, Mark, and Marianne. You can use your emoji, do all the things. I want to see lots of emojis happening. Okay, seeing a few, thank you. Thank you so much for sticking with us a little bit longer today. As a reminder, everyone will get an email shortly in the next day or so when these items are available online, and these items are the webinar recording, the slide deck, and all the resources that we were furiously dropping into the chat. Those will all be in a document, much easier to deal with than a crazy chat. And look forward to more of these discussions. I would love to have a recap of this at some point, you know, before, during the study or after the study. And I think this is also a call for all hands on deck. It is definitely, you know, it takes a village that for decades to get to this place. And if we're going to move this forward into a phase 3, the folks you're looking at on your screen are not the only ones responsible for making that happen. It's all of us making that happen and calling this out and demanding space for on-demand products that are delivered by the individual, products that are not systemic, products that have different levels of efficacy, to make sure that people really do have real choices and real options that they can pick from and are not forced into one sort of regimen or another. Well, that takes all of us making that case and demanding that. So with that said, we're going to officially close this webinar. Thanks again for all of you being here, and I hope to see you when we discuss vaginas and whether or not they demand perfection in a month. And we have lots of other great webinars planned for you in the fall and next year. So hope to see you at all of them. Take good care and be safe.
Thank you.